Excellent.
Yes.
Okay. Yeah, so this webinar series is a project of the Science Transfer Program funded by the Great Lakes Fishery Commission. And our goal is to kind of bring researchers and scientists and technologists and fishery managers together to learn about cutting edge technologies and think about and talk about how they can be applied to management. Just a few Zoom tips before we get started. Please mute your audio when you are not speaking. We will have some time to ask questions of our speakers, and so you can do that one of two ways. You can raise your hand. That's under the React button, that little heart on the bottom. If you click on that, there should be a raise hand option. Or you can use the chat and drop any questions or comments in the chat, and we'll be monitoring that as well. And then I just wanted to do a little bit of background on the webinar series. So we are doing an initial pilot series of 6 approximately monthly webinars during the first half of this year. All the webinars are on Thursdays at 2 p.m. There's a QR code there if you want to visit the webinar series webpage, and also Ted should be dropping some links in the chat. So you can visit that page to register for upcoming webinars, to watch recordings from our previous webinars, and get a little bit more information on the project. The previous webinar recordings are all on YouTube at Lamprey Control. And quick plug for the next webinar. I don't have the registration information up yet because I'm still waiting on the talk titles and abstracts and everything. But pretty soon we'll be posting the information for this. And actually, I think Ted, did I give you the link for registering for that?
Yes.
Okay.
I'll drop it in.
Okay, thanks. Okay, so we do have the registration link up. It's not at the website yet, but on 22nd of May, we'll be featuring acoustic telemetry, some kind of practical tools for analyzing acoustic telemetry data to inform decision making. So we'll be hearing from Tom Binder, Sylvia Ivanova, and Paul Bazanek on that webinar. So that, one last thing. Another link that Ted is dropping in the chat is the link to sign up for the listserv, so you don't miss any of the future webinar announcements. So please do consider signing up for that. Okay, stop sharing here. And I do have just one quick poll before I introduce our first speaker. So because we are attempting to have kind of a dialogue between researchers and managers, just wanted to get a sense for kind of how folks categorize themselves and their role to see who's out there in our audience. So I see the results coming in. I'll give you a minute to respond. Okay. Slowing down. A couple more coming in. Okay. So I'm gonna go ahead and end the poll. Share the results here. So not too bad, mostly researchers, biologists, and we do have a few fishery managers and technologists, and then we have some others. If anybody feels the need to or is willing to drop in the chat what what other means, that would be really interesting to me. Okay, thank you for humoring me on that. So close that off. Oh, policy, good one, Tammy. I'll put that in the next poll. Okay, so then without further ado, I want to introduce our first speaker. So Dr. Margaret Docker is a professor of biological sciences at the University of Manitoba and the lead editor of the Journal of Great Lakes Research. So Margaret, take it away.
Okay, great. Let me share my slide. Okay, yeah, so thank you very much for inviting me. This is the outcome of an STP project on improving eDNA sort of usefulness for resource management. It was a team effort. I'm presenting it because I was available, but involved a lot of work from Chris Wilson, Chris Jurdy, Brian Locke, and Paige Brio, and Julie as our GLFC STP handler, keeping us focused and on track. So just, I mean, I know we're all, I think at this point, at least sort of familiar with the basics of eDNA. It's becoming a really powerful tool for resource management. Usually involves a lot less sampling effort, less specialized equipment. It's less invasive than capture-based sampling, and can provide a really useful complement to capture-based sampling. A previous STP project, we sort of developed a lot of basic resources for use by agencies, by the public, fact sheets, etc., and also a positive eDNA management support tree that if managers got a positive detection, what the possible implications would be. Based on, for example, the repeatability of the sampling, what the likelihood was that detection came from elsewhere upstream or from sort of human contamination. But this assumed that the eDNA results were accurate. It still sort of left a big sort of gap in how fisheries biologists, fisheries managers could evaluate the reliability of the eDNA results themselves. So the sort of both the beauty or the power of eDNA is also its curse in some ways, that because eDNA is so sensitive, it allows for the inference that a species is present sort of sight unseen, but the fact that it's sight unseen, or that sort of invisible man syndrome, also provides challenges in terms of how to interpret the results, because there are, of course, concerns about false positives. That's when the species is inferred to be present when it really isn't, either because of contamination or perhaps because the assay being used isn't sufficiently species specific and it's amplifying something else. And then there's also concerns about false negatives when eDNA might be missing the species if the assay isn't sufficiently sensitive, or if there are various environmental conditions, for example, high flow rates or PCR inhibitors that are preventing detection. So the key for eDNA use is like really, really extensive validation and thorough documentation along every step of the way. eDNA practitioners over the past several years have really been spending a lot of time developing standards for eDNA data acceptance for all of the quality control, quality assurance at every step of the process to make sure that the results are reliable and defensible. Essentially, we have to sort of be our own harshest critics. There have been really emphasizing that validation and the reporting requirements. And within the last few years, oops, sorry, it's not advancing. Within the last 5 years or so, there have been a number of really thorough quality standard documents that have been generated both within Canada, within the US, and internationally as well. What we wanted to do with this STP project, though, was to be able to translate this really sort of dense information that is targeted more for the eDNA experts to the fisheries managers, fisheries biologists for interpretation of the results. So that led to this eDNA decision checklist, where creating an interactive checklist that walked the users through the basic considerations related to eDNA use and the interpretation of the results. And so the goal here wasn't to make fisheries biologists, fisheries managers into the sort of eDNA experts, but rather to have enough information throughout the process to be able to have confidence at the end in terms of what the results mean. So even if the lab portion, the assays, the DNA extraction, the assays is being done by a commercial or other lab, at least to still have familiarity with what goes on during that step and to be able to ask the right questions of the eDNA service providers, I think is a really important part of it. So I'm just going to quickly provide a few words of background, and then we'll walk through the checklist because that's really the high point of the presentation. So the first of the 5 steps was choosing the module. There are sort of 2 basic approaches for doing eDNA work. One is a targeted single species approach, where the assay is designed to pull out and amplify only the species of interest, and then everything else that's sort of that background of eDNA, other species are not detected, or a community or metabarcoding eDNA approach. The success of both of these approaches depend very, very heavily entirely on assay specificity, whether or not you're actually detecting what you want to, and sensitivity as well. And so those will be some of the things that come up in the checklist. The other thing too that I just wanted to mention before we get started, going through the checklist will identify a lot of the really important considerations during that sort of pre-sampling step, but it also really emphasizes how much thought needs to go into the sampling design before you get started. That the sampling effort has to match the question. So, for example, if you're sampling a very rare species, sample effort is going to be required for detection is going to be greater. If you want to have good confidence that a species isn't there, then that usually takes more sample effort than detecting, than confirming its presence and so on. And then the other thing I'll just sort of introduce before we get into the checklist, then as we go through, step 3, the sampling considerations, and step 4, the assay and DNA extraction, there are a number of different considerations that will influence eDNA detectability, et cetera. And these are all things that are going to be sort of identified during the checklist. Okay, so what I'll do right now, I'll just stop sharing the slides here and maybe go to the checklist itself. Making sure that it was working. Are you able to see it? But right now it's probably on the second. Yeah, just a second. I went to the second one to make sure it was working. Okay.
Yes, we can see.
So perfect. Great. Yeah. So here's this eDNA decision checklist, and I think I can say without being embarrassed how wonderful it is because Paige did all of this. It's just such I think a wonderful tool that as I was playing around with it even more yesterday, I'm really appreciating how useful it is, not just for people who are new to eDNA, but even like as we'll see at the end, the report that is generated is really useful as a way of documenting sort of any eDNA work that you might do. So I really encourage you all to take a look at this. So here's the sort of the home page here. So there's going to be the 5 steps that we go through. So you can, sorry, just a second here. I'll just go down. So one of the things that you can either just explore it, or if you put in your name, email, and so on, a report will be generated at the end and emailed out to you, which is really very nice. So at this point here, then you have to decide in that sort of step one whether you're interested in community metabarcoding or that individual targeted species approach. A lot of the considerations are the same in both of them, but there are a few differences. And so that's why you have to choose at the beginning. And you can do this an unlimited number of times for every different project and so on. So then, so that was the first step, choosing the survey type, and then the second component then is the pre-flight check that really focuses on that advanced planning. And so one of the things that's really nice here, I think, is it starts out with a lot of links to existing documents. So we certainly haven't tried to reinvent the wheel. What we've done is tried to select really useful guidance from a lot of these existing documents. As I was saying before, the existing documents are really, really thorough. So we've tried to pull out enough information that it's useful, but not too much that it's overwhelming. And so then this goes through and asks a series of questions, whether or not the project is clearly defined, and you can answer yes or no. Here, for this, I'll say yes. The sampling design reflects the defined purposes, objectives, and gives some sort of leading questions there to help you through it. In some cases, as you'll see here, you can answer yes, no, or not sure. And in many cases, if you answer yes, that's sort of often the end of the line for that question. But in many cases, if you answer no, concerns or considerations will pop up to give you some guidance on if it's a consideration, it's sort of a less, perhaps less essential, but something that you should keep in mind. And then the concerns are a little bit more of a warning, and anything that's in italics here is particularly important. So for example, in the sampling design, the great importance of including negative controls at multiple stages, and so on. And so the same thing here, where you can, there are leading questions as you can go through to think about the things that you should be considering. And one of the things I think is really useful with this is that if you're doing this in collaboration with other partners, that you're sort of organizing the project, and then others are going to be doing the field sampling, others are going to be doing the eDNA lab work. You know what questions to ask of your partners and what documentation that you need from them. And so here, I won't be going through, in the time that we have, I won't go through all of it here, but that as you then, oops, sorry, I went a little bit too far. But here, what I think I'm going to just a second, I finished when I should have gone to the next step. Sorry about that. Talking and clicking at the same time. Let's just see. Okay, so then, yeah, when you answer the last one of one step, it takes you to the next one. And so this is step 3, the sample collection and processing. So again, giving reference to particular resources and particular sections of each of the resources, because some of these documents are quite lengthy, and so identifying page 12, page 13, etc., so you know where to look. And then again, these series of leading questions for things that you should be thinking about, that you should be discussing with your partners. One thing that I really like with this, again, not just if you're new to it, but if you want to use this to track all of your eDNA projects, it also allows you to upload any relevant files. So here is this is the question about the standard operating procedures related to equipment decontamination and sample collection, so that you can upload SOPs associated with each particular step. And at this point, you can upload up to a maximum of 5 files. So so it's just a great way of keeping either your own files organized related to a particular eDNA project, or if partners who are doing the sampling have their SOPs, they can then be linked to this project so that at the end you have all of the documentation assembled in one place. With whether relevant environmental data has been collected while sampling, you can upload up to 10 files related to eDNA collection sheets, the environmental variables that were collected during sampling, and so on. And again, if you're clicking on no, concerns or considerations will come up. The eDNA extraction and assays, again, this is where it can be really helpful if you're contracting with a lab or with a partner to do a lot of the eDNA lab work. Then you can know in advance what questions to ask them. You can know, what are your decontamination protocols? Has the assay been validated? And all those sorts of questions. And again, you can often, if you say no, you get a concern. If you say yes, in many cases, then you can upload the associated files. And then the final step, the interpretation of results. Again, all of the important resources, the raw data. You can upload the files, up to 10 files with the raw data or the interpreted results, again, so that they're all handy in one place, and so on. Okay. I'm aware of the time here, and I know that this is the sort of thing that you kind of have to maybe do yourself. So I'll just sort of go back to a few slides to sort of finish off, and then let Jeff speak. So here, just a second. Let me just see where I left off. I just wanted to show some of the high points of the report then. Okay, and so then yesterday, then when I was sort of playing around, I just sort of generated a report for demonstration here. So this is then a report that was emailed to me afterwards, and so it has, project name, and then it has the answer to all of the questions that I entered are then given to me, so I don't have to be taking notes as I'm working through it. Any of the concerns or considerations are all included there. I sort of think of this as sort of that one-stop shopping, that all of the information associated with any project will be all included. So in addition to the answers to all of your questions, any of those documents that you uploaded, a link to them all is then included. And so now if I want to be able to quickly look at all of the eDNA metadata sheets associated with the project, SOPs, the qPCR results, all of the links are right there. At the end, you get a summary of the number of yes, no, and not sure. There's not any sort of hard cutoff as to what's acceptable. But if you have a lot of no's or not sures, then that's an indication that you need to have some conversations with partners, next steps, additional resources, all of the links are there. And so, yeah, I really encourage you to take a look at this. I think it's a really powerful tool at many stages of the eDNA process. And I encourage you to if you have any questions, you can feel free to email me. Thanks.
Thanks so much, Margaret. Yeah, we have a few minutes if anyone has questions for Margaret. We'll also have some time at the end too, if you think of something. Okay, not seeing any questions. So thanks again, Margaret, for the talk. So our second speaker is Jeff Tyson, and he is the Fishery Management Program Manager at the Great Lakes Fishery Commission. Go ahead, Jeff.
Thanks, Julie. Let me get my screen shared here.
While you're doing that, Margaret, there's a question in the chat. Does the data get stored anywhere?
That's actually a really good question. I would have to ask maybe like you and Paige that in terms of whether, yeah, where I'm, yeah, I mean, I'm guessing it must if those links in the report take me to the files that I uploaded, presumably they're stored someplace for that. And so I don't know if the concern might be sort of data security, or in terms of convenience of how long it might be available. But yeah, I don't know sort of how long it would be stored and who might be able to look at it.
Yeah, that's a great question. I don't remember. I think Paige said something about we do have some sort of metrics on like how many people are using it and are they using it for which, is it species or community metabarcoding so that we can kind of track usage, but I don't know the answer to the question about data storage and access. So that's an excellent question and I will look into it.
All right, you want me to go ahead and jump in here, Julie?
Go ahead.
Okay, thanks. Good afternoon, everyone. I'm Jeff Tyson with the Great Lakes Fishery Commission, and I'm going to talk about applying some of the decision support tools that Margaret just covered very aptly in her presentation. This is sort of a mashup of a couple of presentations that I put together, and unfortunately, as we worked through the Science Transfer Board, Julie had to come up with a different logo for the Technoscience series, but I was strongly encouraging her to pursue the Sprockets theme for the Techno Dance theme for this, but it didn't fly. So first, I wanted to start with a little bit of history. That's my nature, but eDNA has been around for quite a while. I've had some experience in sort of working through some of the challenges with new technology and technoscience, and wanted to cover some of the history of sort of where we've been and where we're at now. So early DNA, historically, DNA had really been deployed primarily in the early 2010s, I guess, in a number of situations. Really, the first eDNA sample collections in Lake Erie occurred back in 2011, targeting bighead and silver carp. These were primarily research or experimental collections that occurred at locations. Let's see if I can find my pointer here. In Western Lake Erie, which is a potential location for colonization associated with bighead and silver carp. So we did some sampling in Maumee and Sandusky bays, highly productive bays, back in 2011. Got results back in 2012, and lo and behold, we had some positive detections. Because this was new science, number one, we didn't know how to react. And in essence, it set off a flurry of reactions, including sort of this initial notification, then communication to other partners, including the Lake Erie Committee, and with partners requesting additional assistance from U.S. Fish and Wildlife Service to try to figure out how to react to it. The agencies then requested another round of eDNA sample collection the year following in 2012. But by that time, we were dealing with this in the media. So we knew at that time that probably we were going to have to put on our Helly Hansens and siren hats and go out and chase some fish around. But we wanted some confirmation from additional eDNA sampling. This slide right here just shows sort of what we were dealing with from a percentage of positives. Margaret showed that in the decision support tree. While not up in that 10 to 70% positive range, still quite high, above 1% of the samples collected. I think there were over 400 samples collected in 2011 and another 400 in 2012. Processed by two different labs. First, the Notre Dame lab processed them in 2011. Processing went to U.S. Army Corps in 2012. But we had a relatively high frequency of positive samples, particularly in Sandusky Bay. There were some QA, QC concerns from the agencies about the results, particularly from 2012. But again, we didn't know what questions to ask, so we had to react. And in that reaction, we deployed crews from Michigan DNR, U.S. Fish and Wildlife Service, and ODNR at a number of locations. I think we had over 300 locations that we sampled over a month time period using electrofishing, blocking nets, gill nets, and all manner of traditional gears to try to capture any big header silver carp that might have been in the system. Fortunate thing is we didn't capture anything, and in the back of everyone's mind there were these concerns. We were dealing with large bodies of water, so 8.25 million acres of Lake Erie. This wasn't a pond, 1 acre pond where you could go out and drain it. So we kind of felt like we were hunting up a needle in a haystack, even if we had positive detections. So that got agencies thinking through time about how to deal with this. And I think that's one of the important messages, particularly as folks develop new sciences out there, to think about other elements that might be worth considering moving forward. This graphic just shows the percent positive eDNA detections for bighead and silver carp in subsequent years. So you can see that from 2013 through 2024, generally quite a bit lower percent positives. U.S. Fish and Wildlife Service took over the processing of the information and really, I think, tightened up a lot of the laboratory protocols associated with processing eDNA, but still some positives out there for sure. And this should be taken with a bit of grain of salt because the sampling, the processing, and a lot of these things have changed through that time period. But this shows sort of the scale of reaction through time and the evolution of sort of what these programs have looked like once they've gotten refined. In addition, once eDNA kind of came on the scene, many of the agencies developed sort of these risk-based approaches, incorporating sort of an eDNA decision tree into a separate fish in hand decision tree that merited different reactions relative to the types of information that were collected. This is an example from Ohio's invasive carp tactical plan. Also, after the initial deployment of eDNA, folks kind of recognized the need to sort of standardize information or communication across eDNA detections and get relevant information out in a timely fashion, and also to provide a readily available record of detections, if you will. So those came along through time as eDNA as a techno tool evolved. And then the last piece is these science transfer tools that Margaret just discussed. She just covered the improving the eDNA reliability checklist, which is extremely useful. And then another decision support tool includes these positive eDNA support trees. So now that we had all these tools out there, we thought there was an opportunity to apply them to a real life situation. So we applied both of those tools to some eDNA sampling that occurred on the Huron River, Michigan, targeted at sea lamprey. Just a little history on the Huron River. There is currently a fish passage project being explored on that system by NOAA, Great Lakes Fishery Commission, Michigan DNR, Huron Clinton Metro Parks, and the city of Flat Rock. And just history of lamprey, sea lamprey in the system. Haven't really had any larval lamprey detected in the system, particularly above the barrier. That's been assessed fairly heavily. Have had some adult sea lamprey detections downstream of 2 dams in that system, but at a relatively low frequency. And so we decided to use eDNA sampling to further inform the risk of fish passage at that location. So it was an opportunity to apply these 2 tools that have been developed. First, Margaret covered the reliability tool, but I wanted to hit on a few sort of notes associated with our experience in implementing the tool. The pre-flight checklist was extremely useful. It caused us to really think about what we wanted to do and where, and then bring in some of that additional information that was out there, like the quantitative assessment surveys that had been done by U.S. Fish and Wildlife Service prior, and it allowed us to document that information. The sample collection and processing considerations were really useful for us because it's super easy with eDNA just to run out and collect additional samples. They're just water samples, right? Anybody can do it, but you need to think about how you're going to do it and how you maintain the integrity of those samples. And this checklist really helped us to do that. And in reality, I think we might have got bitten a bit by this because I'll share some of the results at the end. I'm not sure if it was the sample collection processing or DNA extraction elements, but next slide or next button here, the DNA extractions and assay checklist was really useful because it helped me as a non-geneticist to sort of ask the right questions of the lab manager. And really from personal experience going back to 2011 and 12, I had no idea what questions I needed to ask. We just got the results and it's like, here it is. Here's your positive detections. Thanks. And you walk away. So it really forced us to sort of ask questions in the lab about the processing protocol and those kinds of things in place. And then lastly, the interpretation of the results was another really useful piece in the decision tree here to help us kind of talk with the genetics team about what the results meant and where the caveats might be, which was very, very helpful in sort of getting us through it. So on to kind of sort of the results from the sampling that we conducted, we ended up having 2 positive sites that for sea lamprey DNA from the fall sampling. We sampled both in the spring and the fall at locations above and below the barrier. And then we also had 1 negative control that tested positive for sea lamprey DNA, and that's in the black here. So these 2 sites were positives, 1 downstream of the dam and 1 upstream of the dam, and then we had a positive on the negative control. The positive samples were really quite low. So in this design, we had essentially 3 samples collected from each site. And then those samples were filtered down and 8 technical replicates were generated for each of the individual sites. So we had 24 samples at each site location that were run for DNA. We had 2 positives at this downstream location here, and then 1 positive at this upstream location. All of them were fall samples. One of the other things that we discovered was that as we discussed this with the processing lab, the qPCR machine baseline recalibration, they did one, and that may have led to that positive at the 1 upstream site where the arrow is. So that helped us to better understand what, the positives meant. And again, these were all questions that I say the collective, the Royal We, didn't ask back in 2011 and 2012 because we just didn't have these kinds of tools out there. Next, we applied the management support tree as well. And like Margaret said, the management support tree is based on the proportion of positives in your sample ranging from less than 10% to 10 to 70%. To greater than 70%, and also the vector level, which is an attempt to get at sort of the origin of the DNA, whether it's from a live animal in the system or if it's from transfer of that DNA by other means. A few things that we learned, we actually kind of hit a bit of a roadblock, and I think we can probably do some work on this management support tree to help make it more applicable. But as we move through sort of the tree, most things rely on repeat sampling, and we didn't have repeat sampling. Essentially, we had 1 sampling event, and if we'd gone back in and applied the reliability decision support tree on the front end, I think we probably would have developed our experimental design slightly differently. But I think the value of this management support tree is really in these kind of characterizations of the detection. So it allows you to sort of standardize, all right, we've got some positives, but what does it really mean? And so it runs through a level 1 positive, which categorizes if the target organism is likely not present, but you should keep an eye on the site up to, say, these level 4 positives, where you've got lots and lots of eDNA floating around out there, and repeat sampling that suggests these things are established or at least in the system in high enough numbers that going out with traditional gear makes absolute sense. So we hit a snag here because we didn't have this repeat sampling, so we couldn't ever get to the level 1 or move on. But I think that was one of the lessons that we learned as we moved through applying the tools that we have. So, but I thought they were super helpful, particularly on the sample collection and survey design end of things. It really helped me and the project team to ask the right questions. I think the report was enormously useful. We actually generated that into a technical memo that went right into the feasibility study for the fish passage project. It helped us with interpretation and increased our confidence in sort of what the information meant and informed the messaging for that. And I think the sort of the last thing that, and these are a mishmash of things that came out of this exercise, was with eDNA. Repeat sampling is really, really critical relative to the decision support tree that we have right now, and understanding trend of that over time. And I think going back to some of the trends relative to the early sampling that occurred in Lake Erie and looking at more recent kind of really reinforces that. Cross-contamination, that's always an issue. It's easy to grab water samples and suck them through a filter, but I think you need to be really careful with how you do that, because how sensitive the technique is. It could have occurred on our end at the filtration level, could have occurred in the lab. You just can't isolate that. But when you got a negative control that turns up positive, that should be a red flag. And then the kind of the last point is new technoscience is really, really useful. But I think if you think about new science within the context of how you would interpret and communicate and what that means relative to action, I think you come out much further ahead. It's been over a decade since eDNA has been out there as a sampling tool, and it's taken some time for these other elements to come along, but I think that's what makes the full package. So just a note, not a note of caution, but a note for folks to think as new science comes out, to think about these other elements to make them more management relevant and actionable. But I think that's all I've got. Let's see if I can stop share.
Thanks, Jeff. Great
And
talk.
yeah.
Yeah, we have plenty of time. If folks have questions, feel free to raise your hand or drop them in the chat. Wendy.
So great talk. That tool is really nice, and I'm definitely going to be using one of those graphics or two in the very near future. They're really nice. So I guess just on Jeff's point at the end about sort of as new things come along, I was just kind of thinking about on the query, the first two buckets, you have sort of the two, metabarcoding versus directed assays is sort of two little toolkits that you can use. And probably over the next little while, those toolkits are about to become, a little fuller with different styles of wrenches and screwdrivers, as things like eRNA, LAMP-based, CRISPR-based assays come along, different sequencing platforms. Do you guys have a plan to maybe update this tool to sort of, as these newer things come along, to help people understand what is the best thing to use?
Yeah, Margaret, do you want to take that, or do you want me to get that, or?
I can start, and then maybe you can chime in too. Yeah, I think that would be a great idea, because I know that there is a lot of work being done now to try to get results in real time, rather than collecting samples, taking them back to the lab, and then within a few days, sort of at the fastest, getting results, being able to generate results stream side, particularly if it's in a remote location where maybe action, you want to couple it shortly after detection. Yeah, I think as far as I'm aware, a lot of that stuff is still in the sort of research stage, but as it's coming online so that it has potential for application, I think that it would be a great opportunity to sort of have some tools that would help translate some of those newer tools to potential application.
Yeah, and as far as kind of plan for updating and maintenance over time, so this iteration of the project is actually an update of a project that we originally did in, I think, 2017. And that one was led by Amy Welsh at West Virginia University. And she developed the management support tree, and the graphics and the fact sheets that are available there. And then we went back, we talked with the Council of Lake Committees, which is a body of fishery management agencies in the Great Lakes. And they said, it'd be really great is a checklist that we can, have when we work with the labs. And so that's how this project was born, and the checklist. But as part of developing the checklist, we went back to the old products and did some updating with kind of the newer technology and techniques and methods that are available. So we've already done some updating, and the new tool is associated with the GenFish project, and Margaret knows more about that than I do, but I think the intent is, through the partnership with GenFish and the Great Lakes Fishery Commission, is to kind of ensure that the tools are kept as up to date as possible, given everyone's capacity. So a little bit down the road, a few years, we might look at kind of reopening and updating them.
Yeah, and Julie, could I just jump in real quick too? I've had several discussions with folks, less about the technique and more about sort of the positive detection support tree and how that might be further refined. So number one, ensure you've got repeat sampling, but number two, I think we could do a lot with the vector piece and try to refine that so that it's less of a guess, right, about whether a bird flew in and dropped DNA or something like that, and maybe put it in the context of additional targeted traditional effort in the area, or historical detections, or things like that would make it less of a sort of a swag as to whether it was, transient DNA or from an actual organism that was in the system there.
Right. Well, I'll try not to ask Margaret to do another project for a couple of years. So, okay, Rachel Brown, and then I'll go to the chat. There's a question.
Hi, everyone. Thanks for your presentations, Jeff and Margaret. My name's Rachel at Whitney Genetics Lab. So nice to meet you virtually. My question is based more on what we were just discussing here with updating the tools, but more specifically that decision tree and metabarcoding, which is we're doing a lot of work right now in the Great Lakes, and is coming online, I think, for a monitoring project. So I guess, has there been any thought on updating the decision tree specifically for metabarcoding? And Margaret, you just mentioned too, with timeline, having that information readily available within a couple of days, understanding that metabarcoding typically takes quite a while to get through the lab.
Yeah. So that's a very good point. Yes, because metabarcoding is very powerful in that you can look at a lot of species at once, but you don't. It takes definitely more time, even with a fairly streamlined pipeline for the bioinformatics. Yeah. So then that's a really good question, because I think especially with the decision tree, well, partly because that was first came out, I think, as Julie said, in about 2017. So we were mostly thinking about targeted single species, and for a lot of, particularly for invasive species, it often is that targeted approach. Yeah, so if I'm understanding correctly, then that the decision support tree, sort of modifying that to know how to act with metabarcoding data, it would be potentially different in a few key respects, not just a tweak here or there, and that the time would also be an important factor, because if you're needing to act quickly and it's a month or more till you get the results, I'd have to think about that in terms of whether it's sort of tweaking an existing tree, or whether it would almost make more sense to build from scratch. But certainly something to think about where metabarcoding is becoming more common, or for even if invasive species are your concern, if there are like half a dozen or more that are sort of on the horizon that you aren't specifically targeting, this invasive species or that invasive species, but that sort of broad surveillance for a number of potential.
Uh-oh. Margaret's frozen. Okay, you were frozen for a second.
Oh,
You're back.
yeah.
Thanks, Margaret.
Yeah, thank you. I think that answers my question mostly, but also just wanted to throw out there too that, if we can provide any help with that, or answer any questions you may have as you potentially develop that decision tree, we would be happy to. And also, hear about the things that are important to fisheries manager, like that repeatability, like, oh, is that something that we should be thinking about as we plan the field sampling season, going out multiple times over the year? So good to hear about that too.
Great. Thanks, Rachel. And yeah, the Science Transfer Program welcomes proposals out. So if anybody out there wants to build a decision tree for metabarcoding, reach out to me. We can talk about supporting that. Okay, I'm gonna go. There's a couple questions in the chat. So Charlene asked, and this is, I think, a Margaret question, what type of internal positive controls are used to test for inhibition?
Yeah, so for the sea lamprey project, it was an internal positive control that was duplexed within each assay, so that it was the sea lamprey assay and an internal positive control. I think it was like a synthetic, it was a synthetic fragment that was tested within each assay, which is different than what some people do. In some cases, there's a couple of positive controls that are included on each plate rather than within each assay. So we were at least able to see whether there was inhibition sort of in each and every assay, which did make, well, in general, not necessarily for this project, I don't think there was any inhibition detected in the Huron River samples, but we certainly see differences from river to river in terms of the inhibition. So I think the internal positive control definitely is important.
Thanks. Another question from the chat from Louise. How do you see this technology being used for terrestrial wildlife? Do you think consulting companies will use this in the near future? For example, species at risk monitoring for wildlife assessments.
Yeah, I can hazard an answer with that. I don't know very much about terrestrial wildlife. I suspect that a lot of there's probably a lot of interest in it for terrestrial wildlife. I know there's even work being done now on aerial eDNA from air samples, not to mention other places. I mean, water makes it easy in many respects because that helps to sort of spread the DNA around, but that all sorts of different types of environmental DNA can also be collected in terrestrial environments. I think the biggest limitation for consulting companies to be using it is having the assays and the tools developed and well validated in advance. This isn't with terrestrial wildlife, but with aquatic species. I know that many consultants that we've talked to are definitely interested in applying the tools, but they aren't going to have the resources for the R&D required to develop and test assays. And so I think that once that R&D has been done, there would be interest in applying it, and maybe in the in-between stage, perhaps some potential for collaborations to more thoroughly test them. So once they're developed, then maybe collaborations between sort of the eDNA practitioners or academics and the consulting companies to more thoroughly validate before it can be sort of passed on and to consulting companies to use with confidence.
Thanks, Margaret. And then I see Claire has responded in the chat. She's looking for a paper that comments on this. So thank you, Claire. Other questions? Not seeing any, but I have one. Um, okay, yeah, we got a link in the chat with about airborne eDNA, so check that out. Yeah, so this question is kind of for Jeff, but you know, the definition of technoscience is kind of this dynamic triad between technology, science, and society. And you commented a little bit about like the importance of communication, and you showed some sort of media clips. I don't know if you can comment at all about that society piece. Like, do you feel like in the early stages that the public or stakeholders were kind of pushing for a technology that wasn't quite ready yet for full application? Or I don't know if you can comment on that at all.
Yeah, I mean, so I think it probably was a bit on the premature side because we hadn't developed the capabilities, relative as the agencies relative to the interpretation and communication piece. So I think we should have thought more about that on the front end, and we didn't. So that's why politicians pounced, right? And said, you need to get out there and get this thing out of there with your traditional gears when we knew we were looking at whatever, 8.25 million acres in the needle in a haystack. So yeah, I mean, I think thinking about that as as one of the legs of society, I guess, management, communication, interpretation, those all kind of fit under that third leg that I think requires at least some consideration as it's being developed. Like I said, these tools would have been enormously useful if we'd have had them back in 2011, 2012 to sort of help us to communicate sort of what results meant from the new technology, right?
Yep. Thanks, Jeff. Yeah, Margaret, I don't know if you have any comments on kind of the society piece and how that relates to all this, or
Yeah, I mean, and it's tricky, and I'm really optimistic with how things are progressing now because I would say maybe 5 to 8 years ago, I was worried that maybe eDNA researchers had overpromised, and then there had been some disappointments that had been experienced, either false positives that an assay had been used prematurely, and it turns out it was cross-reacting with something else, and there was all sorts of agency angst that came out of those false positives, and I was worried that it was sort of like, okay, we're done with eDNA. I'm not going to trust it. And so I'm really sort of gratified to see that there is this sort of resurgence. And I think a lot of that has come from the closer collaborations between researchers and fishery biologists and agency and taking into account the policy implications and all of those things rather than just there used to be I certainly felt that there was this disconnect that researchers would develop the tool, sort of get to the proof of concept stage and then stop without that full, sometimes very time consuming, full validation, sort of, that really is required before it can be fully embraced. And so then you need to have that gap bridged between the research and the application better. And I'm certainly starting to see a lot more of that now, recognizing that you can't just design an assay, test it in an afternoon in the lab and publish it and then move on to the next one, that there has to be a very, very thorough validation under all sorts of different circumstances before you can sort of test, apply it sight unseen. Often talking with, members of the public, I liken it to when DNA forensics were first starting to be used to solve crimes and all that. That at first it was not, you wouldn't be convicting somebody based on the DNA evidence until there was sufficient research and testing to know what it means if you say that it was this person. There's a 1 in 10 billion chance that it's somebody that it's somebody else or whatever, but there has to be that very, very thorough validation before it can be adopted.
great. Well, thank you for those insights. And that is our time. So thank you everyone for coming. Thank you so much to Jeff and Margaret for your great presentations. Big round of applause, Zoom applause. And yeah, hopefully you all can join for the next one, May 22nd.
Yep.
Bye, everyone.